Hello, guys. Um, I'm sorry for the delay in bringing this lecture to you. I hope every one of you are doing good. So welcome to our first lectures in the molecular biology series. Like we discussed the last time, we said the first part of our lecture will be actually to understand, the cell itself. I mean, just to get an overview of the cell before trying to understand what really goes on within the cell. Because, like I often say, if you don't get the structure of a building, you won't be able to understand, some compartments of that building and what really goes on within there. So, as part of this series of lectures in molecular biology, I think the first thing we need to do is to understand the cell and try to understand what really goes on within the cell, right? So we are just going to take a little overview of this because you'll have a course next semester that talks about cell biology, where you'll delve more and more into the cell and then get to know about some interesting things that goes on within there. So let's go. Um, so Um, so it's often understood, and generally known that the cell is the fundamental unit of life. So it means that every single living thing has to have a cell, right? Every one of them have a cell because all organisms are made of cells. So these are a group of cells that comes together to perform a specific function, right? To help carry on life. So now another thing we need to know is that the cell is the simplest collection of matter that can live. So it's in a sense that the cell is very simple. If you look at the structures, you look at all of these makeups, these are very, very tiny and simple things, but they are capable of what called making up life. Cell structure is correlated to their function. So a lot of the time, most of the cells are actually related to their function. Let's say, for example, we have the prokaryotic cell and that of the eukaryotic cells. So all of these cells are specifically made to suit their function. Now, all cells are related by their descent from earlier cells. Now, like we said again, all cells are from pre-existing cells. And the way or the makeup of that cell is actually from its descent, from its predecessor. Let's say, for example, the prokaryotic cell comes from a prokaryotic cell, a eukaryotic cell comes from a eukaryotic cell. So that is what they are trying to say here. So we know that the study of cells is cytology, right? For the most part, for biologists to be able to understand the cells, they have to use tools. And some of these tools that are used mainly are called microscopes. And microscope is one of the revolutionary tools that has been used over the years for understanding of the cell. And yeah, the cell is often too small to be seen by the eye. So here is just a picture of a cell, a stained cell with different parts of it. And in here, you can see the nucleus of the cell. So here is just a giant complex material of a cell. Let me get my pointer here. So here is a giant, cell with all of its organelles around it. So this is just an image of a cell that has been stained. And we'll talk about staining just now, okay? It's just that let's go ahead about that. And like we said, the microscope has been a revolutionary tool that has been used over the years to study cells. And one of the simplest microscopes that is known to us as biologists, we know is the light microscope, right? And that is because it is easier to deal with and it doesn't require a lot of resources, right? All you need is natural light. If you get a natural light, a natural sunlight, you'll be able to view whatever specimen you have placed on this stage. So here we have the simple light microscope, right? We have where you visualize, we have these magnifications, and we'll talk about these things. And this is where you place your specimen, right? And this is where the light passes through and hit right in here, and you'll be able to visualize whatever it is you find there. And then we also have these adjustment knobs and so on and so forth. These are different parts of the microscope. The idea here is that cells cannot be seen without microscope because cells are too small to be seen by the naked eye. So for the most part of it, we need microscopes to see cells. And that's not just it. we need to be able to stain the cells and be able to see the different parts of a cell. Okay, now, so the quality of a cell actually depends, or the quality of the image that we see under the microscope depends on key things. Three major things. One, we talk about the magnification. Magnification is how the size of the size of an object or the size of whatever specimen it is you want to look at. So the microscope gives you the opportunity or the chance to be able to magnify, right, to enlargen it. So here we have these different types of magnification lenses. We have 10, 100, and 1000, right, for the light microscope. So you have the opportunity to, adjust that to give you a different view, right? Okay, now in nowadays in technological aspect, we say zooming, right? We call it zooming. How you zoom the object helps you understand or see more detail about it. And another part of it is the resolution, right? A measure of clarity of the image. The resolution gives you how clear the image is, right? Or the minimum distance of two distinguishable points. How two different parts of that particular specimen differs. So technically, or basically in a nutshell, the resolution is just there to give you how clear the image is, right? I often make this joke about the iPhone users and techno users, right? One of them gives you a better resolution than the other. And then we have the contrast. The contrast gives us the difference, the different part of that particular specimen that you're trying to look at. So the contrast is there to show you these different parts. And this is where staining comes into place. Now, here is just an image, a diagram showing, the different parts of, the different sizes or the different, the different sizes of different types of organisms, including humans. One that you can see with your eyes and the other you cannot see with your eyes. So we have the aided and unaided eye. If you look here, from this point up to this point, you'll see meters, centimeters, right? Millimeters, micrometers, nanometers. So what I want you to understand is that beginning from here, coming downwards, the up part is the one you can easily see. The meters are larger than centimeters, right? Centimeters are larger than millimeters, and millimeters are larger than micrometers. Micrometers are larger than nanometers. So the idea here is that things get smaller, and the more they get smaller, the more we need microscopes to see them. So of course, you don't need a microscope to see a human, you don't need a microscope to see a neuron, you don't need a microscope to see a chicken egg. But as time, as we move downward, there are some eggs, like for example, the frog eggs, you need a microscope, you may need a microscope to see that. And what kind of microscope you need is a light microscope, right? It's a light microscope you need to see this. And then as we go down, so what I want you to see here is that the blue line here is for things you can see with your eyes without a microscope. And then this dark blue line here, the sky blue line, is something that you can see without a microscope. And this other blue line here is something that you can see with a light microscope. Okay, and then this purple line here is something that you can see with the electron microscope, because electron microscopes are more powerful than that of the light microscopes. So if we go downward, you see most plants and animals can be seen with a light microscope, right? Most bacteria and mitochondria and the nucleus can be seen with a simple light microscope. But that is not for all organisms. There are some organisms that you cannot see it with light microscope. Instead, you need an electron microscope. But then as we go downward, let's say for nanometer things, all right, organisms or particles that are of the sizes of nanometers, you need you need an electron microscope. You cannot see them with a light microscope. So that is the difference here. Now, light microscopes can magnify effectively to about 1,000 times the size of the actual specimen. So like I told you, they magnify to 10, which is the smallest, to 100, okay, the medium, and 1,000 times. So now it increase the size of that particular specimen or that object 1,000 times, allowing you to be able to see it in more detail, right? That's the thing about light microscopes. Now, various techniques enhance contrast and enable cell components to be stained or labeled. So, in order for us to be able to distinguish or understand what are the different parts of that particular specimen we are looking at, we need to have a technique called staining. All right. Maybe for some of you who know about, a little bit of microbiology, you know what they call gram staining, right? Like they have the the negative gram staining and the positive gram staining, right? So these are different types of staining techniques that are used to visualize the different parts of a bacteria to know, the different aspect of it and to know if it's this microbiology. So most of its subcellular structures, including organelles, right? Membrane enclosed compartment are too small to be resolved by a light microscope. That is why we said some organisms or some parts of an organism can be seen with a simple light microscope, but other part of it cannot. So it has to use what we call the electron microscope. Now, let's say, for example, we have a bright field or unstained specimen. See here, it's difficult to see most of the details of this particular organism, right? But when it is stained, right, you see, you're able to see all of these different parts, the cell, right? The nucleus and the different compartments of this particular object or this particular specimen that we are looking at. So this is the benefit of staining. Staining helps you understand how the shape of that particular cell looks like, what are the different compartments, and then what are the differences and similarities that are associated with it. Okay, so we have another staining technique called phase contrast. Phase contrast techniques shows you the different part of or different phases of a development, different phases of this particular thing. And then we have differential interphase contrast, right? Also called Nomarski. It's another form of staining technique, also different types of techniques. Mostly these things are associated with the kind of dye that is used to stain. We are basically looking at the same thing, the same specimen, but with different staining techniques and different dyes. And then we also have the fluorescence dye to stain. Fluorescent dyes are some of the most powerful dyes that are used to look at neurons and how cells move and communicate amongst each other, right? Imagine without a stain, it will be difficult to look at this. But with the stain, we are able to see this particular cell and all of the fibers and all of the different parts of this particular cell. So these are just different types of techniques. And then there's this thing called the microscopy. Now we already talked about the light microscope. Now let's try to mention a few other microscopic techniques that are out there that helps people visualize different parts of the cell. So the two types of electron microscopes, okay, are used to study subcellular structures. Now, electron microscope use electricity. It doesn't use natural light. That's why they call them electron microscope. Now, we have the scanning electron microscope, also called SEMS, right? They focus a beam of electrons on the surface of a specimen, providing images that look 3D. Okay, and then we have transmission electron microscope, also called TEMS. They focus on beam of electron through the specimen. This one focuses a beam of electron on the surface of the specimen, and this one goes through the specimen. So that's the difference for the SEMS and that of the TEMS. The TEMS are used mainly to study the internal structures of the cells, and then the SEMS are used to study the outer part of this particular structure. So that is the benefit of having these two. And so let's say we have the scanning electron microscopy, all right? What it does is like, we talk about the SEMS, right? Here they give us the outer structure of this particular cilia, different part of, these fiber-like structures that help organisms move. And then we have the transmission electron microscope. We have the TEMS that give us the internal structure. How does it look internally, right? Okay, so cell fractionation takes cells apart, which is also another technique in cell biology that is used to, break down a cell and be able to see the different parts of the organelles. Ultracentrifugation or fractionate cells into ultracentrifugation. Ultracentrifugation is a kind of technique also that is used to break cells apart and then be able to understand the different different organelles that are within them. Cell fractionation enables scientists to determine the function of organelles, okay? Biochemistry and cytology help correlate cell function with structure. You realize every time I mention biochemistry, I tend to laugh. It's because it's a beautiful course. Now, here is a technique that has been used, to differentiate cells, right? We have the tissue right here, we have the homogenization, and then we have the homogenate. So what it did is that it took the tissue and placed it in this particular blender and allow it to homogenize. Homogenization mainly means, mixing it together. After that, then they put it on this differential centrifugation. Differential centrifugation means it's going to spin and differentiate things based on size and weight, right? So as that thing spin, all the heavy stuff will go at the end, that is the debris, right, or the pellet, and then you have be separated according to, if you work at the lab, you know what you do, like to centrifuge the blood, and then you have it separated by different elements. So that's basically what's happening here. So here, if you see, after centrifugation, over and over and over, you come over the pellet that are rich in ribosomes. So if you want to really extract ribosomes from the cell, this is a kind of technique that you'll use. So here we have the tissues, we have the homogenization, we have the homogenate, we have the differential centrifugation. Okay, and the technique continues. So eukaryotic cells have internal membranes that compartmentalize their function. So the internal membranes of these eukaryotic cells have different types of compartments. Let's say, for example, you have a house, and this house have different rooms, right? And this in different rooms of the house or different, have different functions. Let's say the bathroom is there for a reason, the kitchen is there for a reason, the dining hall is there for a reason, the sitting room is there for a reason, and the bedroom is there for a reason. So these are the different types of compartments that we're talking about. They're also there in the cell. Now, the basic structure and functional unit of every organism is one of two types of the cells. We have the prokaryotic cell as well as the eukaryotic cell. All right. Just give me a minute. Okay. Only organisms of the domain bacteria and archaea consist of prokaryotic cells. Now, protists, coming. Okay, sorry. So, only organisms of the domain bacteria and archaea consist of. So only organisms of the domain bacteria and archaea consist of prokaryotic cells, and protists, fungi, animals, and plants all consist of eukaryotic cells. Now, comparing prokaryotic and eukaryotic cells, these are things that we've gone over. I mean, you have learned in your basic biology. The basic features of all cells are they have a plasma membrane, they have semi-fluid substance called cytosol, they have chromosomes that carry the genes, and they also have ribosomes that makes proteins. Now, here is something that you will want to remember, right? These chromosomes and as well as the ribosomes that make proteins, because these are things that we'll discuss in future lectures. Now, prokaryotic cells are characterized by having no nucleus, DNA in an unbound region called the nucleus, no membrane-bound organelles. They also have cytoplasm by plasma membrane. So here's just an example of of a bacteria with different regions within it. The main idea here is to show that this is a transmission electron microscope visualization of this particular bacteria of its different size, right? So this is just a cartoon sketch, and this is the actual visualization of this particular bacteria under the microscope. Okay. So comparing prokaryotic and eukaryotic cells again, eukaryotic cells are characterized by having what? DNA. Remember we talk about prokaryotic cells, right? And then we talk about the bacteria. Now we are talking about eukaryotic cells. Eukaryotic cells are characterized by having one DNA in the nucleus that is bound by membranous nuclear envelope. they have a membrane-bound organelles. The cytoplasm in the region between the plasma membrane and the nucleus. Eukaryotic cells are generally much larger than that of prokaryotic cells. And we'll talk about that. Also, why is that? Because, they have a nucleus. And I mean, they have a true nucleus. And also some of the metabolic processes that take place within eukaryotic cells are much more than that of a prokaryotic cell, right? Now, the plasma membrane is a selective barrier that allows sufficient passage of oxygen, nutrients, and waste to surface the volume of every cell. The general structure of a biological membrane is a double layer of phospholipids. This is all of the prokaryotic cell. So let's see. This is an outside of the cell, and this is the inside of the cell. Remember, we talked about the transmission electron microscopes, right? And the TEMS and the SEMS, right? So here we have, let's just say this is the structure of a plasma membrane. And in here we have the hydrophilic region, hydrophobic region, the hydrophilic region as well. So the outer membrane of the cell, the inner membrane of the cell, and that of, the intermembrane of the cell and that of the inner membrane of the cell. And this is not so important for us for our lecture, but it's just that it's important you get to know this for future lectures. The logistics of carrying out cellular metabolism set limits on the size of a cell. The surface area of the volume ratio of a cell is critical. As the surface area increases by a factor of, it's not necessary. Okay, this one is important and it's interesting to look at. So this is basically a cell, all right? This is the endoplasmic reticulum of a cell, the nucleus of the cell right here. These are the ribosomes, right? They are sites for what? Protein synthesis. We know that some part of the cell takes place some part of the cellular molecular process of a cell takes place within the nucleus, and then the other part takes place within the ribosomes. So this is very, very interesting and important for you to remember as we go through this lecture. So one of the main areas of focus for us as molecular biologists in this class is to know what processes takes place within the nucleus of the cell and what are these things that are there. And another aspect will be what part of this molecular biology process takes place within the ribosomes. So we're not going to go further with this, and I just want you to keep this image in your head as we navigate through this lecture of molecular biology. It will all center around these two areas, one in the nucleus and the other one around the ribosomes. And that was like, a bacteria cell, right? A cell of, a bacteria, and this one is a plant cell. And this is an animal cell, and this is a plant cell, and you can see the different structures. And one thing they have in common that we molecular biologists are interested in, it is the nucleus. They have the nucleus and that of the ribosomes. And that is because both plants and animals have genes, and they need to be able to, synthesize these genes to continuously be able to carry on reproduction and develop new structures and organelles for their survival, for their continuous survival. All right, so you can read the rest of the lecture. It will be interesting and important for you. Like I said, here, these are things we're going to be focusing on. In the nucleus, DNA and proteins form genetic material called chromatin. Chromatin condenses to form discrete chromosomes. The nucleolus is located within the nucleus and is the site of ribosomal RNA synthesis. So on and so forth. So this is just, the rest of the lectures is just for you to read and understand the different parts and functions of a cell. Watch out, you can watch out for the rest of the lecture, and also remember, if there's any question that you have the opportunity to ask that question in the chat room, in our WhatsApp chat room. Thank you very much. I look forward to the next lecture. Thanks.